this. This is where we're at. And show this each time just to kind of keep track where we are. We're probably kind of a lecture behind, but actually we're in good shape. And people have started this STEM Ambassador program, and 2 people have asked me about this. There's another program that I help get going at USM called STEM Sisters. The coordinator is Sarah Chang, and 2 students in this class asked if they could perhaps do STEM Sisters instead of the STEM Ambassador program. And I talked to Sarah about it, and she said that they could. She's very familiar with STEM Ambassadors, so she can design STEM Sisters so that it would be a comparable amount of work. And if you're curious about that, you can contact Sarah, and she can send you information about it. And so again, we have that option on our syllabus to participate in STEM Ambassadors, and there might be a couple of students that get involved in this STEM Sisters program, and that would be great. We stopped on this. This is an important figure, and we'll get back to it. This is where we ended last time, but before we get back to it, I just want to talk a little bit of a review from previous lecture. So we saw this, and again, you don't need to try to read that. It's just another descriptive embryological fact that all animals' initial mitotic divisions of the fertilized egg, the zygote, all of them fall into these patterns. And nowadays, we're not surprised to see that, but the first people in human history that saw it were pretty surprised, you know, because there are usually exceptions to every rule. Why weren't there exceptions to these? Why weren't there some mammalian fertilized eggs that divided in a different way? And we know today, well, it's because of evolutionary relationships between these animals. And in the same way that you're used to thinking about how, well, these are mammals, and they have characteristics like hair, and they have characteristics like womb, and that helps us classify them. And sometimes there can be erroneous classifications because sometimes bodies look surprisingly different than close relatives. The same is true with this really early embryogenesis, is that we have this common plan, it diversifies, and then earlier there's diversification too. But closely related embryos have really similar early development. So we're talking about frogs, and all amphibians have early development like the frog example we're using. There aren't really any exceptions to that. Then we're going to talk today about the chicken, and the chicken represents birds and reptiles. They're all the same like that, and we'll talk some about the mammals. And that's what's going on in chapter 3, is it's talking about vertebrate body plans, setting it up to get to this, and talking about that, oh, they start in some different places depending on the type of vertebrate they are. So we talked through this, and then there's this homework assignment that's due by Friday. So email it to, picture to me, you know, sometime before Friday. And you might do multiple drawings of this early frog development. And it's really the material that we covered for most of the class last period. So you can go back and review that to find stuff. The information's in our book too, but it'd be really great if you used all the labels we used in class, because the book has some of those terms scattered, because it's going through this in a sort of a cyclical way. So it's going to talk about, in chapter 3, it talks about early frog development, then early chick development, then it throws in some zebrafish, then it talks about early mouse / human. And then it sweeps around again and starts talking about them again. And then it sweeps around and starts in chapter 4 talking about a molecular level regulation. And so some of these terms in the book kind of pop up in a scattered way over chapter 3, 4, 5. So if I were you, I would just go, and you don't have to listen to the entire lecture. You can kind of drag the cursor through the YouTube and you can find it, because we're trying to draw and label the development. Does anybody have any questions about that? Or anything at all? All right, on we go. So here's just some more images of that early frog development. We've got this cleavage stage. We've got complete cleavage. That means that cells are forming complete cells. Fertilized egg and early embryo is called a zygote. And in amphibians, the early zygote develops into this blastule, and then it goes through gastrulation and durulation and so forth. Okay? So we zoomed in on that, and if you have any questions about that right now, you can just interrupt. You can also bring those questions to class on Thursday because your homework assignment isn't due until late on Thursday. And so that's the idea behind that. And these are all important critical concepts, central concepts in this course. Here's another image. It's a cross section, and this is a colorized image. And what this is like is, it's like the authors pretending that they've made a fate map. So they're saying, oh, what if we somehow tag this blastula? If we tag these cells with colored dyes, what would they eventually become? And so blue is fated to become ectoderm. That means if you don't interrupt or disrupt or disturb the embryo, these cells are going to become ectoderm. And the big ectodermal derivatives are the skin, the epidermis, and then the CNS, central nervous system. And they're not ectoderm right now. So that's a critical concept in fate, is we can say that this is presumptive ectoderm. It's going to be ectoderm if we don't disturb the embryo, like that. And we could make a more detailed fate map. There's a certain region that's fated to become the central nervous system, like that. Already, it's already. And so this is a fate map. This is the idea that there's this belt of mesodermal cells around it, like the equator on Earth. And this is an image that shows that all three presumptive germ layers are on the surface. And the embryo has some thickness to the cells, but they're considered to be on the surface, and we got to get the endoderm and the mesoderm folded inside to establish the proper organization of the triploblast body plan. And so that's the role of gastrulation. Gastrulation is the beginning of morphogenesis. Here's an early gastrula. The blastopore is starting to dent in, and as it does, these cells are really changing their position, and they're undergoing this convergent extension to make the belt longer, and instead of falling off the front of the body, the belt folds in, and we see this folding in process. So see, here is the little line of the eventual digestive tube, the primitive tube, digestive tube, or God is called the archenteron, and overlying it, we got folded in there these 5 fingers, and this is a cross section, so we're seeing just one of the fingers, and we got the notochord, flanking somites, flanking lateral plate mesoderm. And this is just showing there's the blastocele. The blastocele just gets displaced during development and eventually disappears. And so that's another image of what's happening during development. Here's an image, another drawing from your book, and it's a timeline. And so see up here, we are starting in the early gastrulation. So there's already a dent, and it's already started to dent in. And but we really haven't folded the notochord in. And so the next timeline point is fold the notochord in. And the notochord is already starting to induce this central nervous system, the neural tube. And on the outside image of the gastrula, you can start to see neurulation begin. And so there are these folds that are going to be the neural ridges, and then we have the floor plate of the ventral neural tube that's early called the neural groove. And up on those ridges, see along here, these are going to be the famous neural crest cells of all vertebrates that are really a hallmark feature of the vertebrate phylotypic stage as well. We got some neural crest cells, and no other animals on Earth have neural crest cells except for the chordates, essentially the vertebrates. Then a little bit later, the neural tube is folded, and we got that going on, and we start talking about the position of the somites, lateral plate mesoderm, and so forth. Okay? And so multiple views in the book, you might have multiple views in your little drawing homework. Like that. Here's another thing we talked about, just an image I didn't show. And this is just the classic amphibian egg that you see around here, frogs and salamanders. And we talked about there's this asymmetry. This is just an egg, and mom produced an asymmetric egg, and you see, just with your naked eye, you can see evidence of that asymmetry. The upper part's pigmented, the lower part's light. It's bobbing in the water to keep the upper part up, and that happens because there's more yolk down here. We talk about the animal and vegetal pole. Here's just a little foreshadowing from chapter 4. Here the scientists are looking just at one protein, and they're looking at the distribution of that protein in the mature unfertilized egg. And the protein is tagged with a glowing stain, and so it just shows that the protein is asymmetrically distributed. And there's lots of factors like that we'll talk about in Chapter 4. Chapter 4 talks about and 5 talk about a ton of factors. And in order to take home principles for the rest of your life, we're going to try to distill a smaller number of those factors to learn about them and remember them. And there are a bunch of transcription factors and a bunch of secreted signaling factors. This image right here is a famous, it was one of the, it was probably, I guess it was the first image that people took of molecular factors inside an unfertilized frog egg and saw this asymmetry. And they had experimental evidence for the asymmetry of a variety of different types of things. But here's the first image, and this happens to be a secreted signaling factor. And so mom puts secreted signaling factors in the egg. They're inactive, and they stay inactive, and then during early development they get activated, and they start being released from cells. And finally, we talked about this idea that during this early mitotic cleavage, really the cytoplasm of this great big egg is just going to be kind of proportioned out. And so you can see right here that cells that are in the vegetal pole are going to have some of this factor. In fact, it was called VEG1. And they'll have it, and there really won't be hardly any up here in the animal pole cells. And those are going to be different, and so that's an example of cells becoming different by unequal cell division, mosaic development. So on a test, you'd be like, define mosaic development and give some examples, and you'd be like, well, and early amphibian embryogenesis, the mitotic cleavage, good example. So that's just going back over some of that stuff, and we want to be really solid on it. That's why we're doing that homework assignment. So we'll go back to this. We talked about this figure, and this column is this idea that the concept of fate. And you actually have to do an experiment to establish fate. You actually have to inject the early embryo with some dye, some kind of tag, to be able to see specific subsets of cells and then ask what they become. And so this would be an experiment that scientists started to do in the late 1800s, and they still do it today. But the bulk of the work was done in around the turn of the century, from 1900. Scientists started manipulating embryos, and one thing they did is they made lots of fate maps. There's some little marine invertebrate that lives off the coast of Maine that was really famous because it had some natural pigments in the egg cytoplasm, and they got unevenly distributed. So it was a great early example of fate maps, because you didn't even have to inject anything. There were just some natural differences in the pigment in the cytoplasm, and so they could see, well, what are these cells going to become, like that. Normally we think of it as being a real experimental embryology where we're going to do some injection. So there's that. And then at the end of class we started talking about this, and these scientists started doing this around 1902, and that is they would take embryos and they would move cells around like that. And there are a variety of experiments, and we're eventually going to talk about one, and we'll talk about that in a few minutes, a transplantation experiment between some early salamander embryos. And but here's some terms, and the terms that we're going to learn are so when cells are determined and when they're specified. And so what does it mean for a cell to be determined? And what it means is that when a cell is determined, even if you move it to a different location, it will become what its original fate was. And so here's a cell that's determined. It's up here early in the embryo, and if you move it to a new location, it still becomes what its original fate was. And we could ascribe any fate to this. We could say those are fated to be brain cells, any cells. So this is a generic drawing of all cells, and early cells will have a fate, and then they may be determined. And if they're determined, it means their fate can't change. You can move them to a different place. Why would a cell's fate change if you moved it to a different place? And the answer is that then in their specific embryonic neighborhoods, there are these secreted signaling factors. Cells are talking to each other by secreting the signaling factors, and if a cell is not yet determined, it's still responsive to secreted signaling factors in the neighborhood and can change its fate. That's what's happening here. So here's a cell that's fated, we'll say, to become brain cells, and when you move it, its fate changes. It's not yet determined, it's undetermined. Some really good samples of cells that are not yet determined are stem cells, right? They don't know what they're going to become. Some cells could become the whole embryo. So they are undetermined. These cells have already a fate, but it's not yet determined. And so there would be a point in embryogenesis, a time point where cells become determined. Prior to that, they're not determined. After that, they're determined. And that's that. This idea of determined is said to be operationally defined. And when you say something is operationally defined, it means you have to do something to demonstrate it. We can't know ever if a cell's determined unless we actually do that transplantation. And so that, you know, that's a kind of a difficult term for scientists to use. You know, they'll want to say, oh, this cell is determined. It's not going to change its mind. There's another more general term, and I'll just write it up here, that is commonly used. And they say that the cell is committed. I don't know how many T's there are. And committed means it's like determined, but we're not actually doing the surgery to prove that. So you'll see that used a lot in our book. It'll say, oh, the cells are committed. It's just a little bit looser term, more casual term of the idea that early the embryo has a fate, but it's not yet committed, not yet determined. Okay? Then there's another term here, and it's called specification. And this is another operationally defined term. You can only know it if you actually do some little surgery. And here what is done is a cell is moved I have never really looked at this drawing very well to see if it's very well representing the manipulation. Yeah. So what they're saying is, it's this kind of a odd way of drawing it. But what the manipulation is that you take some cells, you're going to take them from that green region. They're not yet green cells. They're not yet brick cells. And you just put them in a dish. So instead of transplanting them to another location on the embryo, you just take those cells out and you put them in a dish and see what happens. And if they are specified, those cells will grow and they'll become the cells they're fated to become. If they're not yet specified, the cells will grow, but they won't become the proper cell type. And so specification is another step on that pathway from early embryogenesis to the body, and it's just a step where the cells start to know what they're supposed to do. And when you think about that is, you can like do an analogy of these terms with students in college, you know, like that. And so, you know, I don't know, maybe it's kind of tragic to talk about, but each of you has a fate, right? You know, you haven't achieved it yet, you know, but, you know, like some of you are just going to end up in jail, you know, and you're just like, whoops, you know. But yeah, you know, if undisturbed, you're going to go through your thing, and then you're going to become a lawyer, and then you're going to get in trouble, and you go to jail like that, you know. So that's your fate, and it's there, it's out there like that. And some of you are like, I'm going to law school, you know, and there's nothing that's going to stop me. And so those students would be determined, even if their classmates are like, oh, yeah, you know, I think law school, I don't think lawyers are getting jobs, and I think some of them go to jail. You're just like, I don't care about that. I want to be a lawyer. So that's determined. And if you are determined, it doesn't matter what your classmates say, like that. Specification is that if you just got to study on your own, you could go to law school and achieve that lawyerly fate. But usually when people talk about specification, they're saying that in their own, the cells know what they're going to do, but typically they're not yet determined. And so we can put a little timeline on this. And so it goes kind of like this, where the early embryo, so here's, you know, really early, and it's a timeline going that way. And then the cells are going to have a fate pretty early, and they achieve that fate. And then later they become specified. And that means if you put them on their own, they are going to achieve that fate. They know what they're supposed to become. When they're unspecified, they might have a fate, but they haven't even figured out what that is supposed to be on their own. And then typically later, they become determined that they're not even listening to neighbors anymore. They're just going to be that fate. And you could say this for any cell type. You could say it for mesoderm. These cells are going to be mesoderm. Or you could be more specific. These are going to be muscle cells. They're not yet muscle cells. These steps, we'll learn, are all about changing gene expression. And that's one reason we started out with those quizzes about gene expression and genes, is because what's really going on here is gene expression is changing. When a cell is not yet determined, what happens is if you move it, it is going to respond to signaling factors in its new neighborhood, and that's going to change gene expression in the cell's nucleus. So we'll learn that there is a way of how signaling factors could cause cells to become different by affecting gene expression. Yep?
So the difference between specified and determined would be that specified cell is growing like not in a body, so it doesn't get those paracrine signals.
Mm-hmm.
But if you were to put it in like an induction media, it could still recapitulate its fate and become something else theoretically.
Mm-hmm.
Okay.
Yeah.
Got it.
Yeah. And I'll give you an example. So these are really central terms in the history of embryology. And one time, and so for the lab class of this course, we always go down to Maine Medical Center Research Institute because all the scientists down there are developmental biologists and they all study this kind. And one time we visited this guy. He doesn't work there anymore. He went to another spot. He's a stem cell biologist, Joe Verdi, like that. And we went, and Joe is really gruff, like that. Like I was sitting behind Joe at the start of a seminar once, and one of his graduate students came in and handed him a article that they were writing. And it was, probably 30 pages long, like that. And they said, here's a draft of the article, and they were just delivering it to Joe before the seminar started. Here it is, Joe, I got it going, take a look at this. And Joe looked at it, I watched over Joe's shoulder, then he looked at it, and he goes like this. He gets a pen out, and he, God, just when you talk to people like this, you just start to weep. Because he's just like, just on the first page, he just goes, no, start over. The student's just like, like that, right? At the end of our course, you don't get a final grade, and you say, hey, I didn't get a final grade, and I say, start over. What? The student was just crushed. So Joe was like that, and so we went down to visit these scientists, and we went into Joe's office, and Joe was watching a Red Sox game, and he didn't even look up, He's just like, questions? Poor students. He was not going to talk to us at all, only if they had questions like that, And so I knew, that this was not going to go well. And so what I did is I said, well, Joe, I've got a question. And Joe is a stem cell biologist. So he's studying all this stuff that you see about in the news, about regenerative medicine and stuff. And so I said, we spend a lot of time in our course talking about fate and specification, determination, commitment. And he says, as a stem cell biologist, what do you think of those terms and principles? And Joe said, I think they're a bunch of S-H-I-T. Because he said it, and he didn't even look up. He's just like, I think that, And we'll see that we're grounded in these principles, and scientists are learning about patterns of gene expression, and they're learning how to manipulate cells, and in some cases change their fate using new molecular techniques and things like that, right? So could we take a bone marrow stem cell and somehow give it some treatment so that it could make brain cells like that, And so people are excited about this, and so we learn about this as this is the normal and classical understanding of embryogenesis, And when we have those principles, we can actually start to talk about stem cell biology, because there's a lot of that in this course, and understand what's going on in that context. So that's that. Questions anybody has? All right, so here's an example. Here's an example of that. And it's talked about in chapter 1, and the experiment was done about 1920 in Germany. And it's done on some little newt salamander embryos, and it's done on early gastrulae of those. And they've got 2 species. And the species had some different pigments in the cells, so they could tell the species apart like that and keep track of who's who. And they're going to do some transplant experiments. And these are little. So remember, I emphasized that in early frog embryogenesis, the embryos are not growing. They're just building an embryo based on the building blocks that mom put in the fertilized egg. And so those little eggs that you see in the streams around here in the spring are what we're talking about. So obviously this is done under a microscope, and one person cut, little patches out. Now, how did that person, and her name is Hilda Mangold, how did that person orient herself? And what she did is she waited until the blastophore was forming in the early gastrula. And so all of her experiments, or all the really relevant experiments, were done on the early gastrula. And there was the pigmented side to the egg, and then there was just this little blastophore dent, like that. I should just show you this idea, this concept, to help you understand how this Hilda was orienting things. So, here is like, that could be, the earth, or it could be one of these eggs. So it's got this pigmented side like that. And when we look down from the top, so if we look down from above the North Pole, this egg has rotational symmetry. So we talked about asymmetric distribution of ingredients, but the asymmetric distribution is really all along these latitudinal lines, and around it there's rotational symmetry. And so to start out with, this is all equivalent, but as soon as the blastophore starts to form, that rotational symmetry is broken. And we've talked about how the blastophore is going to go in further. It's going to become a deuterostome, so this is going to be the eventual mouth, and basically the anterior end and the posterior end of the organism. This is going to be the left and the right. Okay? We got in this axis, we're going to start to have dorsal and ventral. So just with the denting in of the blastophore, we break that rotational symmetry, and we really start putting together a body plan. So, Hilda started out with that. And and then she used those to orient it. She just removed different regions. And again, you don't have to remember these people's name, but scientists talk a lot about Hilda Mangold because it's thought that maybe nobody else has ever lived who could do that work. Embryologists do phenomenally detailed manipulations. Like, raise your hand if you know Doug Curry. I know Doug. Doug Curry's a professor here, okay? When he was in graduate school, he used to dissect fruit fly larvae under a microscope. And when you do that, Doug would want to open them up and pin them out. So he'd have them in a dish and some liquid, and he'd have a kind of a rubbery surface he was going on, and he'd pin them out, And so, what's he going to use for pins? Oh, Doug, what are you going to use for pins? And Doug would go out and raise your hand if you know what stinging nettle is. So stinging nettle is a plant that lives around here, and if you brush against it, it has little tiny thorns on it that are basically invisible. And they get them in your skin, and you're just really itchy like that, and you're really itching out these things, and you couldn't possibly see them. Doug would go outside, and he'd collect, and and he'd be like walking around looking for stinging nettle, and he'd collect some leaves, he'd take them in under a microscope, he would, with the tweezers, he'd pick off the little stinging nettle, and he'd stand them up in this little rubbery surface, and he'd pin out his little fruit fly larvae like that, you know? Like that, just completely crazy. Here's how crazy it is. Those people, Doug and I know each other because we worked together in the same lab in Seattle. And those scientists in Seattle would do those kind of manipulations, tiny, tiny manipulations. And they would go and just get jacked up on Starbucks coffee to do it. Because their hands would be shaking, because you couldn't possibly consciously move your hand in such minute way. So they would just be under there just hoping, well, I hope this one works, you know, and they'd just be cranking down Starbucks. That's why Starbucks is popular, is because scientists in Seattle are just chucking it to be able to wiggle their hands in this shaky little manipulative way. And those people think that Hilda Mangold was uniquely talented. And then they also honor her because these experiments were done a long time ago where they didn't have, say, antibiotics, they didn't really have a lot of tools, and so most of the experiments failed from just bacterial contamination. So she did this hundreds of times. And we'll just mention that the experiment we're going to talk about, I think it only worked 3 times. Only 3 embryos survived and did this thing that we'll talk about. That's called a positive result, and it's really interesting to compare positive and negative results in science. If you get a negative result, you don't know. If nothing happens, you don't know. Maybe something went wrong, maybe there's a technical problem. A positive result, you just have to see it a couple times, you're like, oh my goodness, you know? I don't know what's going on, but it really happened. And so she just had this work a few times. She was working as a graduate student for this guy, Hans Spemann. And he was already a really famous embryologist in Germany. And Hilda actually went to a talk he gave, and when she was, I think, in college, and said, I want to work with that guy. And another reason people honor her is that there were no women in science at that time. None, like that, you know, essentially. And so, you know, she went up to this guy and said, you know, well, you're pretty cool, I'd like to work with you. And so they worked together. And then it turns out that then she got blown out. Sorry. But people always say that. They're always like, you know, honor, 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 and then she got blown up. You know, like they just, like that. And she was working in her kitchen and just, the kitchen blew up. So she died. And, yeah, no, it's just like, yeah. Like that, yeah. It's like she's like 25 years old, and you're just like, yeah. So anyway, that's the deal with her. And but you'll never forget her. She has a Wikipedia page, and I looked at it, but I forgot to have it to show you, because it doesn't say that in the Wikipedia page, and it's like the Wikipedia page is short. But one thing it says is it's thought that her PhD research was the only work that ever led directly to the awarding of a Nobel Prize, and he got it because she was dead. And you can't, you can't, the tragedy of getting blown up is that you can't get a Nobel Prize when you're dead, like that. And so she did all the work like that, you know, and then he like managed to, you know, and it's this classic sexist thing where he's like, I'm not going in the kitchen, you know, like that. Yeah, okay, so what the deal is that she did these transplants from one embryo to another, so we have sort of a source embryo and we have a host embryo, like a kidney transplant idea. And there's really only one interesting result, one positive result. So in transplanting all these different regions, nothing really happened except what she referred to as the dorsal lip of the blastopore. When she transplanted those cells to the tummy of the host, something really surprising happened. And what happened is that the host embryo produced a second embryo, a second body on its tummy. And so like, you know, it used to be called Siamese twins, now it's called conjoined twins. So twins joined together, and we have an embryo with another embryo growing on its tummy, like that, you know? And they just like, that, you get a Nobel Prize, buddy. That is wicked cool. So it's shown here a little bit. Kind of key discovery that wasn't really shown until after she died, but people already were on this idea that in this second twinned embryo, much of that body is actually the host cells. And so we can talk about with our terms now. This would be some of the ventral ectoderm, and that's faded early to be skin cells, epidermis of the tummy. And when you transplanted this other cells, these other cells on, somehow the fate of the host ventral epidermis changed. And a twin was made, and the cells of the host were being used, including the central nervous system of the twinned embryo was made from host cells. So, you know, it was really dramatic. Like, here's some cells fated to be ventral epidermis, ventral skin cells, but something had happened to them so that they actually change their mind and become brain cells. Like that. So it's early on, and what you'd say is those cells were not yet determined, and they got affected. And so what we'll learn as we go through these chapters is these cells, the dorsal lip of the blastopore, are secreting signaling factors. And so here's that thing, you know, where the belt is getting longer and cells are folding in. And as cells fold in, those cells are in the region of this dorsal lip of the blastopore, and they're receiving signals that are secreted signaling factors, and we'll learn what those are, and that affects their fate. They're going to be fated to become things like the mesoderm, and that includes the notochord. And so what happened is when you move these cells down to the tummy on the host, the secreted signaling factors were made, and they started changing fates. They changed the fates of mesoderm on the tummy of the host, and that included making a notochord. And so a second notochord was made, and that induced the overlying ectoderm to become a central nervous system. And so, you know, we've already learned a lot of the terms and concepts that explain this result. At the time, they're just like, what is going on? You know, this is really interesting. But further experiments led to the ideas that we're talking about, that the dorsal lip of the blastopore cells must be releasing some kind of factor that can affect the fate of the host cells, that the host cells are not yet determined. They're fated to become ventral epidermis, but they're not yet determined, and we could change their fate. This is one of these experiments like this. It's just a little bit tricky, because in this figure we're really thinking about how we transplant cells, and the transplanted cells change their fate. In Hilda's experiment, what happens is she transplants these cells, and the transplanted cells change the fate of surrounding cells, because it's the transplanted cells that are bringing the signal, and they affect the surrounding cells. And so it's just colorized down here that, you know, the neural tube and some of the other cells are actually produced by the host embryo. Like that. And did anybody have questions about that? Oh, you'll see we talk a lot about this dorsal lip, we don't want that, of the blastopore. And it has, the authors use this term. It also has synonymous terms. So people call it the Spemann organizer. Or sometimes our authors just call it the organizer. And so you'll see a little bit different use of that same term, but dorsal lip of the blastopore is a great one because it provides kind of the most information about what it is. Okay, and we'll be especially interested in following that because it can help organize the second embryo by secreting signaling factors. One of the things about that is, so much of the body is made by or induced by the notochord, the somites, and the lateral plate mesoderm. And those are considered to be more dorsal structures. And so what Hilda was doing is transplanting some of that organizing factors down onto the tummy. And that's how they got a second embryo going, is because you know, if you make a notochord, you're going to make a central nervous system. Maybe you're going to make a brain and spinal cord. Well, that's starting to look like a second embryo. Then it's going to have some somites. They're going to make muscles and bones. That's looking like something. Then it's going to make a lateral mesoderm. That's going to make a coelomic cavity, gut, and start making internal organs. So, you know, those players are enough to make a pretty good-looking embryo on the tummy. All right. Does anybody have questions about that? And again, this is an example of this early experimental embryology. It was done in the early 1900s, like that. And then these people ran into kind of a roadblock because they couldn't purify those signaling factors. You know, they could provide experimental evidence, oh, what is it releasing, right? And they just couldn't get to it. Meanwhile, and we talked about this a little bit, meanwhile, in the early 1900s, the field of physiology was moving forward. And I said, and it was literally true, that you can go to a slaughterhouse and get physiological body parts, like the blood. And so you can get buckets of blood really easily. And they would get buckets of blood, and they'd use canoe paddles to stir the blood up in a big old trash can in order to do some of the first purifications of hemoglobin, like that, you know, because they'd just be like, you know, we have really kind of primitive purification tools to try and understand the molecules in blood. So we're going to just use a ton of blood. It's going to be kind of gross. But there's nothing like that for embryos, right? You'd have to dissect little parts of them. And it would be at specific stages that these factors are present only. You really hard to attack that biochemically. So the other field that could try would be genetics. And geneticists were just struggling to set up the principles of genetics. And they would do mutations that would be on physiological body parts like pigment of flowers or, you know, peas and stuff like that. And so it was just no way. So that's how this field became sort of a backwater, where students would take a class in embryology in college, but it wouldn't be very exciting. It would be a lot of just naming and labeling of things like, oh, it's the notochord, it's so awesome. And only in the late 1900s, scientists started to get the tools, both genetic tools and chemical tools, to start identifying these in all different kinds of organisms. All right, great. So that's that. Now, we're kind of done walking through the story of what we can see in frog embryo and in early development. We're going to go back and we'll talk about it again and again, and we'll talk about molecular factors, and we'll try to talk and learn about the molecular factors that get released and why do they get released by the dorsal lipidoblastopore. Remember, there's this rotational symmetry in the unfertilized egg. How do some factors get into that, you know, asymmetric location to play that role? So we'll learn that. That's a bunch of chapter 4 and into some of chapter 5. The authors now, they go on and they describe other vertebrate embryos, and they talk about zebrafish. And zebrafish is a model organism, like a fruit fly or a nematode, and or a mouse. And this book has quite a bit of zebrafish biology in it, and we're going to de-emphasize zebrafish because it's just extra. It doesn't really add much. The reason the book has it is that lots of scientists study zebrafish, and so lots of the people who teach this course around the world are zebrafish biologists, and they would emphasize it. They'd be in their class and be like, ta-da, you know, we're going to learn a lot about zebrafish in here, because I know a lot about zebrafish. And just to mention, well, so anywhere in your book you read zebrafish, it's just not that important. I'm not going to be asking questions about zebrafish on the exam. But that you're assigned to read through it. And because it's a really powerful model organism. And the reason it's a really powerful model organism is a vertebrate, and it has external fertilization. So it's a vertebrate where you can easily study this early developmental events. That's really hard in a mammal. mammals have internal fertilization and early embryogenesis. Internally, you have to kill the mom to study the babies early. The zebrafish has external fertilization. Then it has another really surprising feature, and that is the yolk in the zebrafish egg has to be clear. And so you're really familiar with the yellow yolk of a chicken, and really typically yolk is opaque. And that makes it a little bit hard to see what's happening to an embryo because it has a lot of yolk in it. The zebrafish has clear yolk, and so it's possible to just take a living, normal zebrafish and just put it under a microscope and start watching it. And so people do a lot of like time-lapse videos of zebrafish. And there are ways of using molecular genetics to cause different parts of the early embryo to glow, and you can watch the different glowing parts moving under the microscope in a living embryo. I'll give you an example of that. So my friend Kate is a zebrafish researcher, and she studies face development. So she studies the idea that the neural crest cells are going to migrate up here and they're going to zip together a face. And you might know there's an organization called the March of Dimes, and you might see at a cash register there'll be like put your change in here for the March of Dimes, and that's the original name of that organization, is that they just wanted people to donate dimes of change, and they study birth defects. And Kate gets a lot of research funding from the March of Dimes because she studies birth defects, and the birth defects she just studies are facial deformities in humans, like cleft lip and cleft palate. She's trying to understand that, how that forms and what is happening in that developmental process, and she studies in this fish. So what she has is she has some strains of zebrafish that have been genetically modified so that some of the nerves that grow into the face glow. So we'll learn about that later in this course, that it's possible to genetically engineer organisms so certain cells will glow. And so Kate has these strains that glow, and that makes it really helpful because you can watch in this clear embryo, you can watch the nerves growing into the face, and if there's any disruption, any birth defect, you can see it in the patterns of these nerves growing into the face. And so what Kate and her students do is she uses those strains as a starting point to look and do these genetic screens, like we talked about scientists doing in nematodes when they were studying programmed cell death. Kate does that, and she uses these zebrafish glowing strains to study and look for mutations that have a problem in facial development. And then ask about what's happening with those in humans. So she's trying to identify the different proteins and stuff and genes that are involved in vertebrate facial development. And so the zebrafish is really awesome in specific contexts like that. You can study it really fast. Could you study that in fruit flies? Well, no, they don't have a face. They don't have a vertebrate face. They don't have neural crest cells. So you really have to turn to a vertebrate like that. And this is a really powerful tool for vertebrate embryogenesis. So we talked about frogs and what they look like. We're not going to talk about zebrafish. We're going to talk about chickens. And so here's that same story, starting with an unfertilized egg, and then early embryogenesis in a chicken. And this chicken embryo that's going to form at the phylotypic stage is going to look just like a mammal. And so just to mention that idea that, at first you're like, well, chicken's okay, but a chicken embryo looks just like a mammal at the phylotypic stage. So we look just like that. It's a phylotypic stage. There are no limbs quite yet, right, because they're going to make wings, and so it's early. But at that point, we look just like it. Remember, we had a picture of dolphin embryos at an early stage. We look just like dolphins, and then there's some differences in growth. The same thing with chickens. Chickens look just like us, and other reptiles, birds and reptiles look just like our body, like that. And so we spent some time talking about this because we're really talking about what our bodies looked like when we were really little. Which is a fascinating thing to just dream about, right? There was a time when this whole class could fit in my pocket, You're just like, really? Yeah, I could have beat you up, like that. And it was because you were really little, all right? And so here's the deal with chicken embryogenesis, and it is that the early egg is So the big yellow yolk in a chicken is the egg cell. It's one cell. And mature egg is really big. What's going on there is, I already mentioned, well, they're going to lay the egg out in the environment, and it's going to have to hatch into an embryo, that into a baby that can get up and walk around and eat. And so reptiles have big eggs with a great big egg cell inside of it. And it's going to get fertilized. There's so much yolk that when this early zygotic cleavage occurs, it's incomplete cleavage. And so we can just count, there'll be some images of that. Maybe that's not the best image of that. You can kind of see here that these are attempts to divide into separate cells, and the cells can't fully divide. And so that's called incomplete cleavage. And so what we have is that, here's the big egg cell. And again, there's lots of yolk. And so the cells start to divide, and you see these like cleavage furrows like that to try to divide the cells. But in one little patch, some cells form, and they're really little cells. And then elsewhere, they're just unable to carve that up. This is just going to be just yolk. There aren't any other really cool regulatory factors down in this region. This is just a big old cytoplasmic region of lots and lots of yolk that the embryo is going to form here. And the embryo is going to form here, and it's going to be like a coin-shaped disc that's on the surface of the yolk. You'd never see that in a chicken egg. When you hatch them open, sometimes you see odd things, but you never see an embryo because at a chicken farm, all the roosters get killed because they're mean. And so if the farmer is raising chickens and they're little and one starts to look like a rooster, they just kill them. And so only hens are present, and there's no way that your eggs are fertilized. If you go to sort of a Whole Foods type market, you can actually buy fertilized chicken eggs. They cost a lot more, and some people have the misguided belief that they are somehow healthier or better. So you can actually go find fertilized chicken eggs, but the bulk of chicken eggs are not fertilized. Hens will just lay eggs every day, whether there's a rooster around or not. And roosters are just mean, and they don't taste good, so you just kill them. And so you haven't seen this before. In this early development, there's a little patch. It's a disc, and maybe it's the size of the head of like the circular area of a pencil eraser. Just a little patch. And so here we are looking down at it. So this image is just a timeline looking down at that little patch, and some people call it a blastoderm. A really nice name for it is the blastodisc. And so here we have a different early embryonic setup, and it's because of different amounts of yolk. This is not a blastula, so that's important. We learned about a blastula. A blastula is a hollow ball of cells. And that happens in amphibians, but it doesn't happen in chickens or humans, mammals. Instead, we make a blastodisc or blastoderm, but blastodisc is just a great term. And it's like a coin-shaped flat embryo. And so consequently, there's just some kind of like different like early architecture. We go from talking about an amphibian that's going to be a sphere to a chicken/mammal that's going to be a disc. And so then here's a cross-section of the disc, like looking at it from its edge, just showing on what's going to happen. And so we're in this patch where the cells are dividing and forming in the early embryo, and we get this layered look with cells on top being the epiblast, and those are the significant cells that are going to make the embryo's body. And here's how that process occurs. If you look down, we get this patch, a circular patch, kind of about the size of a pencil eraser head kind of thing, on the surface. If we're looking down at it, we see that there's a little dent. It's like a little wrinkle in like a blanket. Little dent that forms on one side, and the dent folds out into the embryo like that. The dent is called the primitive streak. And the primitive streak is going to be equivalent to the blastopore. The blastopore is the dent in amphibians where the cells are going to start to fold in. And so we're going to get to a point with this development where the cells are moving in to form the triploblast germ layer organization. And so it's going to be gastrulation. And in this case, the cell movement is going to be, instead of denting in, it's going to be like a wrinkle, like kind of like a valley. And cells are going to be sliding in from the valley. And then we're going to learn at one end of the valley, there's a special set of cells that are producing a lot of the body plan. And so we get this formation of this primitive streak, and that's a nice cross section of it, and it's already sort of hinting by making a little colored fate map. It's hinting that cells are going to fold into the primitive streak. Now, what cells will fold in? Well, those would be the mesoderm and the endoderm. They got to get inside and start organizing that body plan. And so here's a nice image of that. And so we've got this primitive streak. It's a dent. And it's going to show gastrulation. So we have a primitive streak, and at this end is something that in chickens is called Henson's node. And in mammals, it's just called the node, which is cool. Here, the drawing is poor representation. Henson's node is actually a bump. It's a little bump, and it's a one end of the primitive streak. See which end is it at? So here we have the disc-like structure, and the primitive streak grows out, folds out like that. And at this tip is the node. And most of the time, I'll just call it the node. During gastrulation, the node moves along from this direction toward the origin of the primitive streak. That process is called primitive streak regression. And primitive streak regression is equal to gastrulation. And that's what it looked like in our bodies, really early. You know, certainly in the first month of pregnancy in our mom, there was a little primitive streak that formed, and then there was this primitive streak regression. And as the primitive streak regressed, the Henson's node progressed along with it. And the way I describe it is, like, imagine you have a long valley, and there are going to be some cells folding in. So imagine they're like a tree-lined valley, and so the trees are kind of falling into the valley. Then at one end, there's this node, and the node is kind of like a glacier moving along the valley like this. And it's covering up the valley. So another way of imagining primitive streak regression is imagining you're a gardener, and you've made a furrow in the ground, and you put some seeds in, and then you can just use your hand to just go like this, and the groove gets covered up, the furrow gets covered up. So that's what's happening. And so let's say this is already underway, and we could draw like a little dotted line that, you know, that used to be the furrow, and the furrow, the primitive streak, and that's getting covered up. So as the node moves along, it covers up the primitive streak. Then we'll see if we made a fate map of this at this stage. Almost all of the mesoderm is going to come from this node. And there's going to be some endoderm and some mesoderm out here. And so the node becomes a real big focus in our body development. When you look at the fate map, the mesoderm fated cells, it's just a little dot in the early embryo. And that's because the cells in the node are proliferating. They're dividing really fast. They're going through mitosis. And so that imagine like the glacier is coming along the valley, and as it's coming along the valley, behind it, it's just the valley got all covered up. The glacier is just going to wipe out the valley. And then imagine on the face, on the front of the glacier, as it's moving forward, the cells are proliferating really fast, and they are sliding down and getting covered up. One way to think about that is imagine you're a gardener and you make a furrow and you slide all the dirt to this end. There's a little mound of dirt, and what you do is you just put the seeds right there, and then you just go like this, and as you cover it with dirt, you're actually putting seeds along the line. And that's what's happening. That on the face of the node, lots of cells are proliferating and they're getting covered up. And so we could draw a little cross section of that. We found a blank page. Maybe there aren't going to be good spots. Okay, so we could attempt to draw a little picture like that. So there's the node, and here's the primitive streak ahead of it. And these are, in this region, there are lots of cells going through mitosis, and they're going to be fated to make mesoderm. And so then we're going to go behind the node, and to show that it's covered up, I'll use dotted lines. So behind the node, there's stuff that's covered up, and these cells that divide get covered up, and they make, so it's going to be mesoderm, they're going to make the notochord, and the somites, and the lateral plate mesoderm. This is going to be the eventual anterior end of the embryo and the posterior end. And so in other words, the cell proliferation in the node is a timeline to produce the body from anterior to posterior. Early on, the cells that are dividing on the node are going to make head-like structures, later more tail-like structures. That's really well evidenced in these somites. And remember, the somites are they're going to get chunked into individual pieces, like a hot dog getting sliced up. And they're numbered from head to tail. And the most anterior somites are some of the first cells to divide in the node at the beginning of primitive streak regression. And so there's essentially like a little timer to make the anterior to posterior body in us. And that's different than the frog. When the frog formed, it was all kind of happening at once. Things were folding together, and it was all happening at once, like the neural tube got zipped up at once. And so there's more positional information in the frog, and in these guys, it's more temporal. Because when this forms, these cells are not even born yet. Okay? And the notochord is going to start to induce the neural tube here, and then later here, and later here. And so the neural tube will look like this, where it'll be zipped together at the head end, and then unzipped here, and then more like neural folds, and then it doesn't even exist toward the tail, because it's a timeline. And so consequently, it's possible to dissect just one chicken and see a big range of development, from neurulation to gastrulation to back here, where that's really not even forming a gastrula yet, in just one chicken. Chick. And the book has that image here. And we're going to have a homework assignment coming up that I'll give you plenty of warning about, where we'll work on making this, because this is what our bodies look like really early in development. and this is just one. It's not like a composite. This would be like just take apart one chick or one mammal, and you'd see all of this stuff happening. And so it's just showing little cross sections and showing the different body parts. And so the red dot is the notochord. See the notochord here, and it hasn't even formed back here yet. Then the notochord is going to induce the neural tube. And so up here, the neural tube is already closing up into a neural tube, and then back here, it's still a neural groove, and then here, it doesn't even exist yet. And this is the head end, this is the body, and that's eventually going to be the tail, like that. So we have this progression of development. Here are the somites just beginning to form this pre-somitic mesoderm, and actually forming some official somites here. Here's the lateral plate mesoderm. It starts to expand and hollow out to form the coelomic cavity, like that. These little bumps here represent some early somatic cells coming together like that. Sorry, I did that backwards. So this is pre-somatic cells, and then these are the cells really organizing. This is early in the forming the coelomic cavity, and later in forming the coelomic cavity in the timeline progression. Or maybe I did it right the first time. That's why we're going to have a homework assignment, is for you to sort out that timeline. And that's that. Oh, there's lots of terms that are too small for you to see, but it has some names like splanchnic, mesoderm, splanchnoplura, and stuff like that. So we already know, or are learning, several of those terms. And that's the image of that body plan. And since we already talked about frog stuff, we already sort of set the stage on a lot of the concepts of like mesoderm and ectoderm and endoderm. And we just have to talk about how it's organized differently because of the beginning. And that goes back to that the idea of that phylotypic stage, where prior to the phylotypic stage, there's quite a bit of differences in the early body patterning of vertebrates, and it's really because of the different egg types and their origin. And so that's that, and there's a lot of information there, and that's why we'll deal with it in a homework assignment that we'll talk about later. But I think that's a pretty good image of the chick. Here's just an image from a different textbook that shows the chick in cross-section, and it's developing like a disc. And what they've done here is they've just pretended to take a drawing of it. Instead of having it flat disc, they curved it down like this, to help show in cross-section how a chick embryo is starting to form a body that's quite similar to a frog embryo, just that it's a disc versus a sphere. And this just shows, some of those major features. Talking about the somatopleura and the splanchnoplura, and in here it says some of the splanchnoplura is going to be the cardiogenic mesoderm, so it's going to make the heart. And same body plan, just different starting points. So there's that image, that idea again. So then the next thing to say is that's again what our body looks like. And so when the authors start talking about mammals, they don't talk about that at all. They just say it looks the same as that. And then what they start talking about is in mammals, the embryo produces the body, and it also contributes to a lot of the extra embryonic membranes, like the placenta. And so when your mom gave birth to you, she gave birth to your body, and then later delivered the afterbirth that includes the placenta and the amniotic sac and so forth. And your cells were in the afterbirth also. The placenta is composed of some cells from mom and some cells from you. And so the book wants to talk a lot about what's called the extra embryonic cells. So we got the body, you got the other cells that you and I contributed to during our development in our mom's tummy. That turns out to become a wickedly complicated story. And so the book tries to do it, and so I think I have an image here of the book trying to explain what it's like when the mammalian body plan gets put together. And here they're talking about a mouse, any mammal like us. And you just don't have to try to figure that out, because it's just complicated how all of that folds together. The way to understand the body is the body looks like just like a disc, just like the blaster disc of a chick. Imagine that you take a disc and you just kind of fold up the edges, you'd have a bowl. And it's true that in the early mammalian embryo, it develops as like a bowl-like shape. It's a disc, it's a flat disc, but it just has to be curved up. So it has just a little different look to it. But on the surface of that disc, in the epiblast, those cells, we get the body plan. And then beyond the disc are all these other cells, extraembryonic cells. And so I have, we'll go back to that, I just have this figure from a different book, and it says, let's learn 3 terms from early mammalian development. And so these are new terms, and the terms are inner cell mass, trophoblast, and blastocyst. We want to learn those terms because they are players in some of the story of how mammalian embryogenesis is different. The mammalian egg is tiny. It doesn't have any yolk, and it doesn't have any of those those kind of building blocks like amino acids and nucleotides, because it's going to implant in the uterine wall. And so we're going to learn about the early development, and it's different. So with the frog, we had the early complete cleavage, rapid mitotic divisions dividing up the big, mature egg into portions, and we formed a hollow ball of cells, the blastula. Then that doesn't happen in chicks. Chickens have a really big egg, and so they just make incomplete cleavage, and they just make a disc-like shape. Really early in mammalian development, that really early events aren't like that. In really early, we just have a 1-cell zygote, and it just divides to make 2 cells, 3 cells, so forth, and it's called a blastocyst. So we have blastula, blastodisc, blastocyst, early, like that, forming. And we'll see that the blastocyst gets organized into an outer layer of cells called the trophoblast, and an inner layer of cells called the inner cell mass. And so we can just kind of go like that. It's a timeline. Here's mammalian fertilization, and then we're going to have a 2-cell, 4-cell embryo, and it's going to make a solid ball of cells called the blastocyst, and then that solid ball of cells is going to become organized into an outer trophoblast and an inner cell mass. And we want to learn those terms because it's just a really different organization than the early chick or the early frog. Also, we'll see that there's really interesting stuff happening in the mammalian embryos at this stage that is unique. So we learned these 3 terms, and then just see this, that the trophoblast is going to give rise exclusively to extraembryonic cells. In other words, it's the inner cell mass that's going to make the embryo, the body. Even the inner cell mass contributes a lot of extraembryonic tissues here. So the inner cell mass is going to give rise to the embryo and extraembryonic tissues, the trophoblast, extraembryonic tissues. Then here's the embryo forming the primitive streak, blah, 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 blah. And so that's why we learned those 3 terms, is because of the organization, and it's the inner cell mass that's going to give rise to the embryo. And just as a last image to show you something that looks familiar, here is a mammal, and it's releasing an egg, so ovulation, and then the egg is moving down the fallopian tube, and that's the location where fertilization typically takes place, and we start to have a zygotic mitosis. We get a 2-cell, 4-cell, 8-cell, 16-cell, and it gets to be a blastocyst, and that's what is going to implant in the uterine wall. Here's the idea that that early development, the primitive streak happens in sort of a bowl-shaped region instead of a flat disc. No big deal. Here's that bowl-shaped region like that, and then lots of extraembryonic cells starting to form the connection, the eventual placental connection with mom. you know, eventually forming the placenta and umbilical cord and so forth, amniotic sac, the embryo grows in. We got to stop there, and we'll keep going. Remember, if you have trouble